Welcome to lecture number 12.
We are still in module number 2, talking about cell biology and histology.
Our learning objectives for this lesson are going to be 2 bullet points of the nucleus and protein synthesis. We're going to talk about the structure and function of the nuclear envelope, nuclei, and chromatin. We're going to talk about the genetic code, gene, transcription, and translation. And we're going to talk about the cell cycle. So the cell cycle, you've seen this in middle school and in high school biology. So we're going to cover that. All right, so our cells. Our cells contain genes. The genes are the unit of heredity. They contain the instruction codes to make DNA. The key part of the information transmitted from the parent cell to the daughter cell. And these are found in the nucleus. All right, the nucleus is the command center or control center of the cell. The nuclear envelope is the membrane around the nucleus. The nuclear envelope has pores in it, these little holes. And the little holes, the pores, are selective, have selective permeability. So they have selective things that can enter and exit the nucleus. The nucleus has ribosomes. It has ribosomes because it also continues right into the rough endoplasmic reticulum that also has ribosomes. And this chromatin in here, this contains 30% DNA, 60% proteins, and 10% RNA. All right, so the cell cycle. The cell cycle has 3 parts. Interphase, which is the longest portion. This is where the cell grows and carries on its normal activity in interphase. Then it's going to go through the mitotic phase here. And the mitotic phase is where we get the division of genetic materials. After the mitotic phase, it's cytokinesis. So this is where the cell divides into 2 cells. So we have interphase, which is the majority of the time the cell is in interphase. Then we have the mitotic phase, which kind of just goes from prophase, metaphase, anaphase, and telophase. That's kind of the 4 that's mostly in mitotic phase. Then kind of throughout here, you're going to start seeing cytokinesis happening. And we're going to see that in a little bit. All right, most cells need to replicate continuously for growth and repair. So we're going to show you how that happens. So mitosis is the division of DNA in chromosomes. First phase is prophase, which is this first area right here. In this area, the nuclear membrane dissolves and the chromatin thickens. Then we go to metaphase. The chromosomes line up on the middle shelf. Then we go to anaphase. The chromosomes are pulled towards opposite ends. And then we go to telophase, where the nuclear membrane reappears. 2 nuclei with 46 chromosomes each are in it. And then cytokinesis is where they're going to break apart. So let's talk about interphase. Interphase is the major phases of the cell cycle. It's interphase. This is the longest phase. Interphase is the period from cell formation to cell division. So when a cell carries out its routine activities and prepares for cell division, that's during interphase. So as you can see here in this picture, we have the nucleus, the nucleolus, the nuclear envelope with the pores. We have chromatin in here in the middle. It's uncondensed chromatin. Then we have some centromeres, or centrosomes, excuse me, with centriole pairs here. And then out here we have the plasma membrane. So the subphases of interphase is G1 or gap1. What happens in there is there's a vigorous growth and metabolism. So cells that are permanently ceased dividing are said to be in G0 phase. Okay, when we figure out we need more cells, they'll go to G1 and they'll start making more DNA so that we can start getting ready to divide. The next is S synthesis. So S stands for synthesis. This is where DNA replication occurs. During the S synthesis phase, to make identical copy of the DNA, so each resulting daughter cell has a full copy of genes. G2 is gap2, and that's where it prepares for cell division.
So let's look at this summary of DNA replication. It starts out with chromosomes that end up being in this double helix, and we have old DNA. The first step is the uncoiling. This is where an enzyme here will unwind the DNA helix and expose the bases. The separation is happening at the replication fork. So this is where the hydrogen bonds between the base pairs are broken and the DNA strands separate right here. Two new strands, a leading strand and a lagging strand, are synthesized in opposite directions. The leading strand is synthesized towards the replication fork, and the lagging strand is synthesized away from the replication fork. And the replication fork is the point at which the strands unzip, right here. So, in the assembly, we have an old or parental strand that acts as a template for synthesis of a new strand. Okay, so it's going to take that, act as a template, and it'll make a new strand. This strand is called the leading strand, and it's formed continuously, and the lagging strand is formed discontinuously. They're synthesized in opposite directions because the polymerases work in one direction only. So the DNA ligase enzymes splices short segments of the lagging strand together. Then the DNA molecules recoil. So after DNA replication is complete, two identical DNA molecules are formed from the original. This process is called semi-conservative replication because each new double-stranded DNA is composed of one of the old strand and one of the new strand. During DNA replication, histone proteins, which are made in the cytoplasm and imported into the nucleus, associate with the DNA, forming the two new chromatin strands. These condense to form sister chromatids. So these are sister chromatids here, and they're united by a centromere. Now these are going to stay held together up until the anaphase part of mitotic cell division. Then they're going to get distributed to the daughter cells, and that's going to make sure that the daughter cells have identical genetic information. All right, so as we talked about before, mitosis involves prophase, metaphase, anaphase, and telophase. You can remember that by remembering PMAT, P-M-A-T. So the first step is prophase. In early prophase, the chromatin condenses and it's forming these bar-shaped chromosomes. Each one of these is duplicated. Its chromosomes consist of two identical threads called sister chromatids, like we saw in the last slide. These are held together by a centromere. As the chromosomes appear, the nuclei disappear, and the two centrosomes separate from each other. Okay. The centrosomes act as focal points for the growth of the macrotubule, assembly called the mitotic spindle. As the microtubules lengthen, they push the centrosomes towards the opposite poles of the cell. The microtubule arrays, called asters, extend from the centrosome matrix here. All right, late prophase is where the nuclear envelope breaks up. So now we don't see the nuclear envelope. This allows the mitotic spindles to interact with the chromosomes. So now the spindles are interacting with the chromosomes. Some of the growing spindle microtubules, called connector microtubules, attach to the kinetochores, the special protein structures at each chromosome's centromere. The remaining spindle microtubules that are not attached to any chromosomes but function to force the poles apart, okay, because they're going to be pulling it to each side of the cell. Now, you only need to know about prophase. The early prophase and late prophase, you don't need to know those separately. The only reason it separates them here is because they kind of look different when you look at the pictures. All right, the next one is metaphase. As metaphase starts, the two centromeres are at opposite poles of the cell. The chromosome clusters along the center of the cell. So they're going to be in the middle, metaphase middle, with their centromeres aligned along the equator of the spindle. Okay? This imaginary line midway between the poles of the cell is called the metaphase plate. Enzymes that will separate the chromatids from each other are triggered at the end of metaphase. Okay. Anaphase, this is the shortest phase of mitosis. It starts abruptly as the centromeres of the chromosomes simultaneously split. Each chromatid now has become a chromosome on its own. The kinetochore microtubules move along by the motor proteins in the kinetochores, gradually pull each chromosome towards the pole of the cell that it faces over here. The moving chromosomes take on the shape of a V letter. The centromeres lead the way and the chromosome arms dangle behind them. Okay. And the last phase, telophase. Telophase begins as soon as the chromosomes stop moving. This final phase of mitosis is like prophase in reverse. There are now two identical sets of chromosomes located at opposite poles of the cell. Each set uncoils, and the chromosomes resume their thread-like chromatin form. Okay, a nuclear envelope forms around each chromatin mass. A nuclei reappears within the nuclei, and the mitotic spindle breaks down and disappears. That's the end of telophase. Now, cytokinesis, this is going to begin at late anaphase. You're going to start to kind of see a little bit of cleavage. But during late anaphase, the stage of mitosis continues through and beyond telophase. You're going to start seeing cytokinesis. During cytokinesis, a contractile ring of actin microfilament forms in the cleavage furrow. Eventually, this furrow starts to pinch the cell apart, which will result in two daughter cells that are identical to each other and their mother cell. All right, there's some additional resources on this. If you look at page 100, there's a focus figure 3.4. You can also play this little video about mitosis, and you need to make sure that you review what's happening to the nucleus, the nuclear membrane, chromosomes, centrioles, microtubules, centrosomes in each phase. All right, control of cell division. Control of cell division is controlled by internal factors and external factors. The ratio of cell surface area to cell volume. That's what limits how big a cell can be, and explains why most cells are microscopic. Chemical signals such as growth factors and hormones released by other cells. The ability of space. Okay. Normal cells stop proliferating when they begin touching. This is a phenomenon known as inhibition. Okay. So they stop dividing when they're going to start crowding each other, when they start touching each other. Over here, we have some restriction checkpoints. So checkpoints are key events in the cell cycle where cell division processes are checked. And if faulty, they stop there and they get repaired before they move on. So the G1 checkpoint, this is a restriction point. It's the most important of the checkpoints. If the cell does not pass, it enters GO or G0, in which no further division occurs. All right, meiosis versus mitosis. So here in this slide, you can see mitosis and meiosis. We haven't really talked about meiosis much, but we need to know the difference for this test. So, as you can see here, mitosis is over here, meiosis is over here. As far as DNA replication goes, in mitosis, it only occurs during interphase and before mitosis begins. In meiosis, it only occurs in interphase before mitosis 1, but not in mitosis 2. Okay, number of divisions. Mitosis only has one division. Meiosis has two. Make sure you know that. Synapsis of homologous chromosomes does not occur in mitosis. It occurs during prophase 1. Tetrads form and allowing crossover to occur. Crossover is where the genetic material mixes. That's only going to happen in meiosis. Number of daughter cells and genetic composition. In mitosis, we have 2. Each diploid cell is identical to the parent cell. In meiosis, we have 4. Each haploid cell contains half as many chromosomes as the parent cell and is genetically different from the parent cell. So meiosis, we're getting genetically different, and in mitosis, it's identical. Roles in the body. So we use mitosis for development of multicellular adult from a zygote. So this is your embryonic growth. Tissue repair and renewal. So like if you get a sunburn, a cut, it's got to repair it. It's going to grow the exact same cells. And it ensures constant genetic makeup of all body cells. Where in meiosis, This produces cells for sexual reproduction. So this is going to produce the gametes. It introduces genetic variability in gametes. So that's from the crossing over. And reduces chromosomal number by half so that the fertilization can restore the normal diploid chromosome. So that's why you get half of your genes from your mother and half of them from your father. So once again, here's that chart blown up, and here's the differences. And that's where we'll stop for these subjects. And if you have any questions, let me know.